Greg Foltz. I'm a neurosurgeon at Swedish Neuroscience Institute, and I direct the Center for Advanced Brain Tumor Treatment.
My name is Madan, and I co-direct lab with Greg. I performed the genetic analysis of tumors and looked at the gene expression changes and the epigenetic changes that drive the tumor formation.
And I'm the neurosurgeon, so I remove the tumors from the patients and bring them up to the lab. I've always been committed to taking care of patients with brain cancer. Actually went to medical school specifically to take care of patients with brain cancer. That doesn't just mean that I do the surgery and look after them afterwards, but also because it's an incurable disease, I'm committed to coming up with new treatments, and the avenue for that will be exploration in the lab.
We are trying to determine what changes in transcriptional activity using RNA sequencing, and also determining if epigenetic mechanisms contribute to tumor formation.
Now, under the microscope, all tumors look the same. But when you look at the genomic level, there are tremendous differences in variability between patients. And we believe that these differences also will correlate with how patients respond differently to their treatment and also the progression of the disease. We look at DNA, we look at RNA, and we look at the modifications to DNA and RNA that affect a process such as cancer. I have not seen a single technology developed that will move us forward as rapidly as the solid system, simply because on a single platform, for the first time, we can investigate every single aspect of genomics in a in-depth fashion at a magnitude that was not available before.
It is like comparing the analog information which we used to get earlier from earlier technology to a digital information. Earlier technologies lacked that sensitivity. For example, let's say if a gene was expressed in 5 copies and in tumors it becomes 10 copies, it's still a twofold change in expression. But using the earlier technologies, we could not detect that change. With the solid system, we can detect that change and understand the biology behind it.
The solid system is going to allow us to get a picture of the entire genome in an individual patient's tumor. What we first need to do is build a database that will give us a picture, a complete genomic picture of each patient across hundreds of patients, and then correlate those genomic changes with the behavior of the disease.
We will be able to investigate the biological networks which are perturbed in these diseases and use targeted therapies for these patients.
I think that at the end of the day, if you're a clinician and a scientist and trying to make a difference in a disease such as brain cancer, it's really about the results. And I can tell you that we have never failed using applied biosystems technologies in being able to get the answers that we need to make a difference in the lives of these patients.